Look at the structural resilience of a high-tensile tendon under intense physical pressure. Compare that to a liquid droplet of olive oil or the rigid, upright stalk of a plant. Macroscopically, these structures appear unrelated, yet they are constructed from the exact same microscopic ingredients: carbon, hydrogen, oxygen, and nitrogen. The mechanism that bridges the gap between 4 basic atoms and the vast range of mechanical properties found in life is polymerization. It is the process of taking small, simple repeating units called monomers and linking them together into massive, complex chains. The physical properties of these massive chains are predictable. By understanding a few strict rules of molecular geometry, electrochemical polarity, and bonding, we can determine exactly how a molecule will behave inside a living cell. Mastering these rules explains the architecture of life. It also provides the specific mathematical framework required to detect, isolate, and quantify these microscopic structures in a laboratory. A water molecule has one oxygen atom bonded to 2 hydrogens. Electrons are shared unequally, giving the oxygen a delta negative charge and hydrogens a delta positive charge. Opposite charges attract, so neighboring molecules align, creating a network of hydrogen bonds. Individually, a hydrogen bond is weak, but millions of them grouped together require a massive amount of thermal energy to break. This gives water a high specific heat capacity, meaning it absorbs substantial heat before its temperature rises. It also results in a high latent heat of vaporization, allowing organisms to cool off efficiently through sweating without losing large volumes of liquid. This collective attraction is known as cohesion. Cohesion allows water to form continuous, unbroken columns that travel up the microscopic xylem tubes of plants. It also creates surface tension tight enough to support the weight of small insects. Water's polarity dictates its chemical behavior. It creates a stable thermal buffer against rapid temperature spikes and provides a highly effective solvent for the biochemical reactions that keep cells alive. Moving to solid structures, we start with carbohydrates. The simplest forms are hexose monosaccharides, single sugar rings containing 6 carbon atoms. Glucose is the baseline model. Because it is covered in polar hydroxyl or OH groups, glucose forms hydrogen bonds readily with water, making it extremely soluble. Alpha and beta glucose differ only by the hydroxyl group on carbon 1. In alpha glucose, it points below the ring. In beta glucose, it points above. When 2 alpha glucose molecules collide, their reacting groups detach to release water, forming a 1,4 glycosidic bond. The reverse of this process is hydrolysis. To break that glycosidic bond and separate the sugar rings, the cell must spend a water molecule to sever the oxygen bridge. Condensation reactions assemble specific pairs of monosaccharides into disaccharides. 2 glucose molecules yield maltose. Glucose and fructose create sucrose. Glucose combined with galactose forms lactose. If the cell continues linking alpha-glucose molecules, it builds large polysaccharides. Plants synthesize starch by creating an unbranched, tightly coiling helix called amylose, alongside a branched structure called amylopectin. Animals store alpha-glucose as glycogen, which contains frequent 1,6 glycosidic branches. Those multiple exposed ends allow enzymes to rapidly detach glucose when an animal requires an immediate energy supply. Beta-glucose presents a geometric roadblock. Because the carbon 1 hydroxyl points up, 2 beta-glucose molecules cannot align their reacting groups normally. To successfully form a 1,4 glycosidic bond, every alternating molecule in the chain must flip physically upside down. Because the units alternate, the resulting polymer cannot coil. It forms long, completely straight chains called cellulose. These unbent chains pack closely together, allowing thousands of hydrogen bonds to form between neighboring molecules, creating incredibly strong microfibrils. A plant's easily digested energy reserve and its indestructible cell walls are both polymers of glucose. The macroscopic physical difference between them is dictated entirely by whether a single hydroxyl group points up or down. Lipids assemble under a different set of rules. They are not true polymers made of repeating subunits. A standard lipid is built from 2 distinct components, a single glycerol backbone and up to 3 hydrocarbon chains known as fatty acids. A triglyceride forms when glycerol bonds to 3 fatty acids, ejecting 3 water molecules. Saturated fatty acids contain only single bonds, keeping the chains straight. An unsaturated fatty acid contains a double covalent bond, introducing a physical kink that forces the chain to bend. Because triglycerides lack charges, they are entirely nonpolar and hydrophobic. They repel water. This makes them effective biological insulators, dense energy stores, and waterproof coatings for animal fur and aquatic bird feathers. Replacing one of those hydrophobic fatty acids with a negatively charged phosphate group creates a phospholipid. This modification yields an amphipathic molecule, a molecule with a water-repelling tail and a water-attracting head. In water, phospholipids spontaneously align their tails inward to avoid the liquid, forming the bilayer that surrounds every living cell. Cholesterol, a distinct lipid known as a sterol, slots directly into this structure. It is mostly hydrophobic, but possesses a single hydrophilic hydroxyl group. This specific geometry allows cholesterol to wedge between phospholipid tails, regulating how fluid the cell membrane remains. Swapping a single hydrophobic hydrocarbon tail for a charged phosphate transforms a simple water-proofing fat into the primary architectural boundary of cellular life. Proteins operate as biology's nanomachines. Their monomers are amino acids. Every amino acid shares a universal backbone consisting of an amine group and a carboxyl group. What distinguishes the 20 different amino acids is a highly variable side chain, called the R group. Condensation reactions link the amine group of one amino acid to the carboxyl group of another, forming a peptide bond. The exact linear sequence of these linked amino acids is called the primary structure, and it is strictly encoded by an organism's DNA. Hydrogen bonds pull the chain into its secondary structure, spring-like alpha helices, or flat beta-pleated sheets. In the tertiary structure, hydrophobic R groups cluster centrally, charged R groups form ionic bonds, and sulfur-containing R groups fuse into disulfide bridges, locking the 3D shape. Many functional proteins require a quaternary structure. This occurs when multiple folded polypeptide subunits assemble together. Some also incorporate non-protein prosthetic groups, like the iron-containing heme group bound inside hemoglobin. If the hydrophilic R groups face outward, the result is a globular protein. These are water-soluble spheres, like hemoglobin, insulin, and lysozyme. Their folded geometry creates specific active sites and receptors, allowing them to target other molecules in the bloodstream with high precision. Fibrous proteins like collagen, keratin, and elastin are dominated by hydrophobic amino acids. They do not fold into soluble spheres. Instead, they form tightly twisted ropes with massive numbers of crosslinks, creating durable, insoluble cables for tendons, skin, and bone. A protein's ultimate mechanical capability is determined by the linear sequence of its R groups, which dictates how the chain folds into a functional structure. Because these molecules rely on defined chemical bonds and specific side chains, we can exploit those features to detect them in a laboratory. Standard qualitative tests target these structures. Iodine visibly darkens by physically wedging itself inside the helical coils of starch. Biuret reagent chemically detects the presence of peptide bonds in proteins, turning purple. The emulsion test precipitates hydrophobic lipids out of an alcohol solution, turning the liquid milky white. Testing for simple carbohydrates requires a closer look at electron transfer. All monosaccharides and some disaccharides are categorized as reducing sugars. This means they are capable of chemically donating an electron to another molecule. We use Benedict's reagent to detect them. The solution contains blue copper 2+ ions. When heated, a reducing sugar donates an electron to the copper ion, reducing it to copper 1+. This reaction forms a brick red precipitate. Sucrose is a non-reducing sugar. It will not donate an electron. To detect it, you must first boil the sample with dilute hydrochloric acid to hydrolyze the glycosidic bonds, splitting the sucrose into reactive glucose and fructose. After neutralizing the acid with an alkali, the Benedict's test will successfully trigger the red precipitate. A heavy precipitate yields a deep red hue, while a low concentration produces a green or yellow mix. The limitation of the Benedict's test is that human vision can only interpret this color gradient semi-quantitatively. Comparing test tube colors is subjective. To extract exact measurements, the procedure must move from qualitative observation to objective optical quantification. Once the red precipitate is filtered out of a completed Benedict's test, unreacted blue solution is left behind. A colorimeter is an optical device used to objectively measure the intensity of that remaining blue liquid. Look at the internal physics of the tool. White light passes through a red filter before hitting the sample.
We use a red filter because red is the complementary color to blue, ensuring a blue solution absorbs red light significantly. If the solution is extremely blue, very little glucose was present, and the liquid will absorb a high percentage of the red light. Conversely, if the solution reacted with a high concentration of glucose, it loses its blue hue, allowing the red light to pass straight through to the detector. The colorimeter provides an absorbance value, but not the actual glucose concentration. To bridge that gap, you must perform a serial dilution. This involves taking a stock solution of glucose and diluting it with distilled water to create a carefully staggered range of known concentrations. By running the Benedict's test on every known concentration and recording their individual colorimeter readings, you can plot the data points onto a graph. This establishes a standardized calibration curve. This chart reveals the concentration of an unknown sample. Take the absorbance reading of your unknown test, trace it horizontally across the y-axis until it intersects the calibration curve, and drop vertically to read the exact concentration on the x-axis. Biological complexity is the result of molecular geometry scaled up. Understanding these bonds lets us reverse engineer biological architecture and extract quantifiable data from its foundations.